Because they tend to melt. They also have silicon properties in them, and then the machine can get it very attracted, and it can. That's the reason. Okay. Yeah. Apart from this form, there is a screening form which has exact details about questions. For example, if they had fallen in the past somewhere when they were 2 to 3 years old, like every detail is there inside that screening form. Which, because the participant, when they come, they might not remember that do they have something inside, because we also scan old age participants over here, so then they tend to forget. So this particular form helps you to be very sure of the fact that they don't have any metal implants inside their body, because it's your sole responsibility that you are taking a participant inside the scanner. So this is
maybe any instrument or something that we scan and get to know.
Yeah, that is the instrument that would also check if something is inside your body.
We can rely on it.
Yeah, definitely you can rely on it.
I have one question. Like
Yeah.
this demographic, does it affect number of slices?
Does it what?
Affect number of slices, or what factor do you determine that maybe this patient have to take this maybe 64 slice?
Can you just repeat your question?
I'm saying you have taken these demographic features
Okay.
for the subject.
Sure.
And like if you're taking this maybe function MRI or structural MRI, we have this number of slices.
So
Yes.
slices is a different thing. Look, it's like we
Yes.
put in the
Yes.
scanner.
Yes.
That is not matched with a demographic thing.
Okay. Like what factor do you determine that maybe for this patient, the subject A maybe have to take 64 slice, maybe for subject B.
No.
That
So when
what are the factors?
Sure. So when you set the protocol inside the host computer, then only you specify what number of slices you are asking the machine to take.
It depends on the research
Okay.
question. And how.
So it is very same for every participant.
Suppose someone has bigger brain
That is what I was coming to. So that is what I was coming to. If the brain size, if the head size, that is a technical term.
The number of slice.
No. Okay.
For this maybe the size of the brain. No.
No, actually no. So the head size doesn't say that you will have more number of slices. What is happening is your head size, if it's larger, for example, that obviously can have number of higher slices, but in the protocol we have specified that it has to cut the brain into say 164 or say 72 slices for every participant. Okay, I'll also tell you there is this 000 reference point. Now how do we compare if you have a higher head size, I have a smaller, and different 000 is the reference point which is same for everybody of us. Okay, and that particular reference point, with that reference point, everything is matched for every participant, so that our data can actually be exactly overlapped upon each other.
For a single research student, we already defined the predefined protocols.
And
for every subject, so we can't change the number of slices.
And when you'll see,
so the number of slices is constant for the
Yeah,
machine.
No, not for a machine, for a particular reason.
For a research, for a single research, for a single research, we already predefined the protocols, and in the protocol, we already predefined how many slices we want according to the previous literature of that state, of that your research question.
Okay, come. ये लो. ये तो हमारे हैं ना, तुम लोग इतना मत करो. इतना मत करो ना, अभी मुझे वो भी तो देखना है. डेट ऑफ बर्थ बता जल्दी से, जल्दी करो. डेट ऑफ बर्थ बता दो. Okay, so this is the host computer. Host computer saves all the demographic details of the participants, and we always make sure that we don't enter the name of the participant over here. We enter an ID for the participant so that the identity is anonymous for the participant. Okay. Chalo, next step बताओ. Patient position. Patient position. So these, as you can see, are the patient positions, which mean head first supine, feet first supine. These are basically the different types possible in which you can make the patient enter. Right now we are doing a brain scan, and that is the reason that head first supine is what we need right now. Right? Next. Sure.
और दूसरे लिस्ट जो है उसका क्या? उसका मतलब ये जैसे मान लीजिए कोई सीधा लेटता है
उसको कन्वर्ट करके लेटना है, तो उस पर क्लिक करने के बाद हम यहाँ डिसाइड कर सकते हैं। हाँ,
समझिए नी का जैसे होना है तो नी
तो
पहले
उल्टा
जाएगा
रहेगा।
तो फिर उल्टा
हाँ,
जाएगा।
तो उल्टा
हाँ जी।
रहेगा।
लेकिन कॉइल फिर नी की होनी चाहिए
नहीं,
जैसे।
कोई दिक्कत नहीं।
सर उसका इधर रहेगा
जिस तरह
हाँ।
जैसे पार्टिसिपेंट जैसे अंदर जाएगा उस तरह से उसको हम डिसाइड कर सकते हैं। कन्वर्ट लेटना है तो लेफ्ट लेट ले, राइट लेट ले।
हाँ
हाँ, यस
क्योंकि
यस
ब्रेन कॉइल ही है वहाँ पे। Okay, so now you can see every body part is mentioned over here. Whichever body part you want to get the participant scanned for, you click over there. So right now we're selecting brain, as you all can see. ये तो सीमेंस की पूरी सब ये प्रोटोकॉल्स है हमारे, ये हमारे यहाँ प्रोटोकॉल्स
हम
है।
लोग बनाते हैं।
हाँ
बनाते क्या है, मतलब बच्चों का एक्सपेरिमेंट्स बताएंगे।
नहीं बताएंगे।
Okay.
Resting the protocol is suggesting this data can be used for assignment for analysis.
Sure.
So make it 7 minutes resting.
7 minutes ही resting, right?
नहीं, 5 मिनट का
Yeah.
7
कोई बात नहीं, हो जाएगा।
हो जाएगा।
7 मिनट का
मेरा 7
Sure.
कोई बात नहीं, इसी को बढ़ा देंगे।
हाँ 7 minutes। Okay.
तो अभी ये पार्टिसिपेंट अंदर डालते हैं, फिर प्रोटोकॉल समझाते हैं।
ठीक है ठीक है। First we'll take the participant in, then I will make you understand how we set a protocol for a research study.
आप जाओगे?
अभिषेक जा।
We have to be here.
हाँ जी सॉरी सर, यस सॉरी।
Have you been
As I told you, we'll also be making you see a task-based analysis. So that's the reason that they have asked him to hold those particular 2 response keys. What you can see there is noise cancellation headphones. So there's a lot of noise that is produced by the machine, right? And it's very disturbing for the participant. So that is the reason that we try as best as possible how much we can reduce the noise. That is made possible through those headphones. Okay, and okay, where is that person? You had asked about that question, right? The reference point. So now you will see, you'll see a red light flickering just on the top of that participant's head. When it will be switched on, you'll be able to see. We match that particular red light for every participant so that it actually gets to that 000 axis. You will be able to see. What he's putting is the most important thing of this scanner, of the scan getting done. That is head coil. Head coil is only responsible for carrying all the signals from the participant's brain to this host computer where we see the image.
through head coil or
Right. See that red light you can see over there? Shashwat thoda aage aa gaya. Right?
Yes.
So they'll be matching it for, it has to come in the center of the participant's forehead and then they'll be taking him in.
So that's
No, that we manually do. Okay, you see a mic over here. This is for us to communicate with the participant, or if the participant is feeling any uneasy. Come.
So we speak anything, we can.
Yeah, definitely. Security button दिया था ना?
हाँ नहीं दिया ना
ठीक है. There is
Security
also a
button नहीं
security button that you hand over to them in one of their hands, that if they feel uneasy at any time, they can press it, and then this thing will start making a noise and we can take the participant out. It's almost never it has happened, but it is always a good practice wherever you do this. हाँ, okay. Sir, तो ये so now we'll be telling कि how do we set the protocol. First of all, your protocol is very, very specific to your own study. Okay, this protocol might or might not be the same for any of us, right? It comes from a lot of research review for your particular study. Okay, so basically you have a research question in mind, and if you know that fMRI can only answer that question best, then you read on your papers over there, and then you come down to what are the exact values for parameters like number of slices, field of view, right, T1, TR, TE. So these are all technical terms that you, whenever you read any paper on MRI, you get a paragraph which tells about data acquisition parameters, and then that talks about these particular values. So you to take those values and then you put it over here. Now you can read more about these values and what do they actually signify, but then you will have to go into a little bit of physics for that. So yeah, but still we can tell that field of view, for example. So basically, which brain area are you wanting to capture, right? Which is your area of interest? So that would define what is your field of view, right? Then there are TR and TE. So time of repetition and time of echo, right? So that depends upon the number of cycles that the machine is taking for the next signal, for it to give the command for the next signal to come. So that is a time of echo. Okay, then there are number of slices, at how many number of slices do you want the participant's brain to be cut into, right? Then you see this is the voxel size you mention over here. Sir, आपको दिख रहा है? जैसे कि यहाँ पे है कि voxel का size क्या है, right? Then resolution, then SNR is very important, signal to noise ratio. So you have to see that you have set the parameters such that the signal to noise ratio is not very high, right?
So we need to fix it.
Huh?
SNR we need to fix it.
So you have to show them maybe cartoon movies, or have their mom go and stand next to like hold their leg or something and ask them to be quiet. But still, like I would say, I would lose maybe 50% because they. That means would be a better option, but. Little better. Sanjeeva,
Yes.
do you suggest eyes open or eyes closed?
Sir, I suggest eyes closed. Because with eyes open, we have seen visual activation as well. They try to see what there is. What do you suggest, sir?
No, I asked you. With eyes open, do you worry that someone may fall asleep?
With eyes closed.
Eyes closed.
Yeah, but since it's a small resting state, like 7 minutes, so
It's okay.
for that I need not worry. But if they have task, so in task, so anyway, the eyes would be open. There is this paper which talks about how restful is the resting state scan actually, right? So
There is another paper. Is it time to put rest to rest?
yeah.
I cannot go inside.
Yeah.
Right.
Claustrophobia.
But there is some paper which shows that there is a difference between eyes open and eyes closed.
True that. That's the reason, and it's recommended to have eyes closed, right?
Right, but don't you? For me, it's either is okay, but don't you think if you want to compare with a task,
Huh.
which where the eyes is open,
True.
then for rest, you should
Sure, that makes sense. Because you need to have a true baseline for.
Yeah. But either, I think either doesn't make much of a difference.
That's good to hear.
So did you scan all the 100 subjects?
Yes, 112.
What we did with patients?
So that is the main issue over here. But we just go and then we persuade them that you will be getting a scan done, which is.
How do you find patients? How do you recruit them? Like word to mouth or
Huh?
doctors, direct contact?
No doctors actually in my case. But yeah, word of mouth.
What do you concentrate on which particular? What do you concentrate on the
Which particular age group?
disorder like this?
Huh?
Which particular disorder?
Oh, no. Mine is with healthy subjects. So I don't have any disorder
What do
participants.
you research on that?
My research is on cognitive and affective aging, wherein I'm trying to see that how emotion regulation, that basically how we are able to regulate our emotions, how that helps in.
इधर start कर रहे हैं task बिल्कुल समझाएंगे.
Okay, so we are starting with the task-based fMRI now. Okay, Shashwat will guide you through it. Batao.
First, this is just the instruction screen. उसमें whatever is the instructions for your experiment, you'll give that. In my case, I have a motor learning experiment. So you can see 4 circles here, right? The black circle comes randomly in one of the locations. It is actually in an order, but the participant will not know it. It has a 3 triplet rule. It has a triplet rule which will determine where the third location goes. So this is an implicit learning task. We don't tell them that it comes in an order. Later on, once the entire task is done, we ask them if they have experienced any kind of order in the sequence, and we also have like a free generation of the entire sequence if they can do it. And there are other measures and other tests for determining if they have explicitly gained knowledge or was it just an implicit knowledge. Now, how do you come to know whether it's an implicit knowledge and it was not explicit? Is that we see the reaction times of the participant when he's pressing all the keys, and what we have seen is that with every block or with every presentation, the reaction time is decreasing. So the reaction time can only decrease when the participant has been habituated with the sequence. But they, so in case of, in this experiment, once he comes out, yes, once he comes out and I ask him whether he has experienced any kind of sequence going on or if he knows the entire sequence, he'll be saying no. But if I have it for let's say like 40 minutes, then probably he'll get the sequence. So there are different factors which can influence this explicit knowledge or the gain of this knowledge. So that's the thing.
What cognitive process?
This is implicit learning.
Just implicit.
Learning, implicit and explicit. So we have a factor which is influencing whether it will be implicit or explicit based on a behavioral experiment.
Here, implicit means what?
Implicit is that they don't know that they know the sequence, but they actually know the sequence, which we can see from the reaction times. But if you ask them, then they wouldn't be able to generate the sequence, or even if you give them a test, they won't be able to predict where the next location of the circle is going to be. But based on the reaction time, they know where it's going to be. That's implicit. If they can produce the entire thing, if they can predict the next location, that is explicit knowledge. So once they press the key, in this case, there is a 1 second gap. So that we call as response stimulus interval. So what we have observed through behavioral experiments is that if you have the response stimulus interval of 1 second, then they gain the explicit knowledge. In his case, he might not, because it's going. This is only a demo session, so it will go on for 5 minutes. My task is for 35 minutes. So after 35 minutes, maybe he has gained. I don't know if he'll gain it within 5 minutes. It depends from participant to participant. He might gain it. And the other condition in which we have seen only implicit learning and no explicit gain is when the response to stimulus interval is 250 milliseconds. So we have these two groups, and then one on the basis of the behavioral experiments, we are hypothesizing that the first group is an implicit group and the second group is an explicit group. Once the data comes, then we can see it. So this is for seeing. Also, sir had talked today about block designs and event-related designs. In my case, you could say that this can be an event-related design since we only care about like the activations which is happening when he's pressing the button. But since I have a learning experiment, what I need is that even during the response to stimulus interval, the learning process is going on. So if you have a sustained activity, which is in my case, you'll have to use a block design. So for me, my block goes on for 18 seconds. There is an optimal duration for every block. In the long group, sorry, in which he is doing, this block is going on for 36 seconds. So there's a 36-second block, a 12-second rest period, again 36-second block, again a rest period. And I have 4 blocks of this, which he'll be doing. After the 4 blocks, he has a control block. Now the control block is acting like a baseline for the other 4 blocks. So it's a 4v1 situation. Now the control block, I have him only pressing one key for the entire 36 seconds. That's it. And I'm counterbalancing it between the 4 locations because after the entire experiment is done, I'll be anyways averaging out all the control blocks. So it needs to be in all the 4 locations as well. So that's the experiment. 35 minutes is for like the entire experiment. This will go on for like 2 minutes more. You can see it from here. Yeah, 2 minutes more. No, only the task. It would, that's why we recommend not going above 35 or 40. But we have had people do it for 1 hour also. You can see from the reaction times. So that is an interesting point because the reaction time would increase, right? So yeah, that has also happened. Yeah, you can see. I mean, in my case, though, the stimulus is coming for 500 milliseconds. If there's no response, that means he is either omitted only that sequence, or if he's sleeping, he's going to skip a lot of trials anyways. Oh, in the resting state. Okay. Resting state. If the participant is sleeping, you wouldn't know it unless and until like the participant tells you. So go according to self-report, I guess. You can't determine it now. Oh, if you want like analysis wise, you can just check the correlation, like the DMN correlation. If it's like very high, then he's sleeping. So DMN will be only for meditators or if he's sleeping. If it's high, then he's sleeping. DMN will not activate if that's not there. DMN is default mode network. No, it wouldn't be. If they're sleeping, you'll find that. I don't have experience with this. I'm not easy. So in that alpha peaks would be yeah. But some people doesn't exhibit it. Like that, it's this network. Okay. Also, there is exclusion for some people. Like that I'm asking. So the peak also wouldn't be there. It'll be like very less amplitude. You're saying. That is there here also. Same thing. Yeah. That is there here also, but if the participant is awake, then DMN will not show. Same thing, like same thing as EEG. Same. In some people, alpha peak won't be there. Yeah, same thing will be there. So DMN activity will be there. The correlation or the value will be very less. No. Like some people, even if they are resting, alpha peak won't be there. Like that would DMN also. Even if they are sleeping. Yeah. Okay. Like that's a new thing. Yeah. I don't think it will be there in our case. DMN will show up. Okay. Yeah, it will.
So yeah, I have one question.
Yes.
So every MRI machine, like you can have structure MRI, function MRI, or there's some specific machine. Maybe this MRI maybe based on all the structure, and this one maybe function, or maybe video scientific can combine structure.
Yeah. True.
So I'm asking, it should be like every MRI machine they can do the same way. Yes.
Every MRI machine can do it. Yes.
It's just MRI. You want to use it as structural or functional. You can use like the scanner in some medical facilities. You can convert that MRI. Yeah. It's not converting. It's just same machine. Basically.
Yeah, it's same machine. You just need to get the components. That's it.
So I said this functional.
Yeah, this is a normal.
There is no separate functional MRI. Just MRI.
The machine is called MRI, magnetic resonance imaging. That carries out your structural MRI and your resting state, for example, as well as your task base. Now, since you are running a task base, that is why
that
it's a
becomes functional MRI. That's it.
That's it.
So it's not like there is a separate machine.
Yeah, the technique is called fMRI. It's not the machine.
Yeah.
Machine is
So maybe there's some other features.
No,
which
no,
also-
it's just the components which you'll need. So the triggers you'll need, the cord, and then the component.
The same basic.
Yeah.
Yeah.
Academic same use.
The participants see the task amount.
Oh, he can see. You can see the screen from there, right? So the screen is, I think, so it's there. The it mirrors this, and the screen is reversed also. So yeah, the screen is reversed. So basically the head coil which you have, that has a mirror like this, and then you can see through the mirror. You can see the screen through the mirror.
No screen.
No, no, no. There is no screen open. Yeah, it's from the reflection. Yeah, that screen is inverted.
Okay.
There. So it's mirroring it, but it's inverted.
Inverted screen and
Yeah.
see the proper.
Yeah, nothing else. That monitor, and also the refresh rate of this monitor should match that monitor. That is very crucial. Otherwise, it won't do it. Yeah, you have to sync that. Yeah. Any other questions? Fine.
Okay. Thank you so much. So now another batch should be ready.
Screening. Do you have a screening form? Screening form. I'll just.
You can
Pata
see
nahi
these functional images.
Yeah. I'll ask. I wouldn't. Laptop में है. अच्छा, screening form. I have it. Joshua, where's the other screening form? Wow. Oh shit, I still have it. पकड़ के चलते थे.
No, but just do it. I don't know.
This is the used one.
Yeah, this.
Yes.
You can show them. You have to wear the recording.
Yeah. पहले बताने दो what it is, how it works. Please come. That's all, right? Yeah, okay. So first of all, this is the fMRI machine. As you know, it does two types of scan. One is structural and one is functional. Structural scan lets you see what kind of problems are there in the brain or the normal structure of the brain. So you can actually see if there is any kind of anomaly like injury or tumor or any kind of infection. And if you want to go for the functional thing, that is, suppose like a patient or a person is showing erratic behavior, irrational behavior, you can actually see what kind of brain function is associated with that particular erratic behavior. Also, if you just want to study a particular behavior, if you want to study the risk and reward mechanisms of a depressed patient, you can just see the scan and study their behavior neurologically. So that is the two types of scan we can do. Another type of scan that is possible is DTI, diffusion tensor imaging, which basically tracks the development of the white matter tract. So we can see the plastic changes in the brain as a function of training, as a function of aging, as a function of normal development as well. So these are the three things we can do in fMRI. Now the other thing is, how does this machine work? As you have already heard a lot about bold, right? So what is bold? Bold is basically blood oxygenation dependent level signal. So this machine does not directly capture your neural activity, rather it is based on your deoxyhemoglobin and oxyhemoglobin level. So whenever we perform some kind of task, whenever your brain is thinking, there is we are using excess oxygen than what we have consumed. So our oxygenation or oxyhemoglobin is more and deoxyhemoglobin is less. Now what happens is when you go inside the machine, the deoxyhemoglobin has paramagnetic property, and as you can see, this is a huge magnet. So it has magnetic susceptibility because magnetic field is varying, right? So whenever you go in, if you have higher oxyhemoglobin and lower deoxyhemoglobin, in that case your paramagnetic property of the entire like your brain signal is reducing. If you have less paramagnetic property, then you will have less magnetic susceptibility and your signal to noise ratio will improve. That is what we capture. That is why we are able to capture. This is why we work with bold signals and not with direct neuronal activity. And now another thing is, how does the entire system works? How are we capturing signals? So once you go in, what happens is it's a huge magnet. So you have lone hydrogen atom in our brain, in our body, like whatever you are scanning, right? So those very small like lone hydrogen atom has very small magnetic dipole. They will get arranged in the direction of the strong magnet, right? So what do you capture? They are all arranged, so there is nothing to capture. That is why we send these radio frequency pulses which will flip those like magnetic dipoles by 90 degree. That distortion is what you capture as the signal. When you capture bold signal and the hemodynamic response function build up, is that is what you are capturing. दिवाकर सर एक बार प्रोटोकॉल उनको दिखाइए, मैं I just want to explain. ठीक है, so I'll just any protocol, कुछ भी कोई भी एक protocol बस खोल दीजिए so that हाँ उसमें कुछ भी है, मतलब TRT थोड़ा दिखाना है ना. Okay, ठीक है. So just in case, I'm just telling you, just remember. So whenever you are working, you will come with words like TR, TE. So just remember what these are. For example, TR is the time of response, TE is the time of echo. What is time of response? So like I told you, we are sending radio frequency pulses and you are getting back a signal. The duration between when you send the radio frequency pulse and get back the signal, that duration is the time of like time of you can say echo. Okay, so it's basically echoing back, right? Now we have TR, which is time of repetition. Time of repetition is, so you are not sending one radio frequency pulse, you are sending a series of radio frequency pulses. So the duration between two radio frequency pulses is the time of repetition. Okay, so these things you need to take care of because you want to maintain a good signal to noise ratio. So obviously it is based on whatever literatures you are studying on the mechanism or work you are doing and what they have suggested. So you try to fix it in your system, and obviously it will not come exactly that, but it can come close to that. Just try to make sure that whatever literature you have studied, your signal to noise ratio overall should not change like a lot. Okay, and we have slice thickness. So you can have very thin slices, you can have thick slices. So there is a trade-off between slice thickness and the you could say the spatial resolution. If you take very thin slices, your spatial resolution will be better, it will be more detailed, but the acquisition time will get longer. Okay, and if you have thick slices, obviously you will not have that much detail, but your acquisition time will be small. Yeah, so that also can affect your signal to noise ratio based on whatever you are studying. Another thing you will have is FOV, which is field of view. Field of view is basically the region you are studying, you have interest in. For example, suppose you are studying motor cortex and you want to look at the internal capsule basal ganglia kind of region, right? So that is your field of view. Okay, now it can be your whole brain or it can be a particular region you are interested in. That should be based on what you want to study and which region is of your interest. So that is your field of view. Again, if you like, you can go for whole brain, but mostly in times when we are studying a particular mechanism, you should focus on the region that is associated with that mechanism, which is already theoretically there. So that is field of view. And slice thickness I have told you, TRT I have told you. I think other than that, there is something called, if you look at the protocol, you will see something called head first. Why? So see, head first is not a like hard-bound rule in MRI. You are scanning the brain, so you need it head first, right? But not everyone needs it head first. So someone can do for lungs and they will go from the, yes, they will go from the back. Yes, fit first. So that's all. And yeah, Oh, there is one more thing called flip angles. Flip angle is basically, I have already told you that those weak magnetic dipoles are getting flipped by the radio frequency pulses you are sending. So the flip angle by default is set to 90 degree because that is what the flip usually happens. But if you have any question where you want to flip the angle by suppose 120 degree or suppose less than 90 degree, that is your question, but that also will affect your SNR, signal to noise ratio, because the moment you like flip the angle, you are making a change in your in the lone hydrogen atom volume, or you can say density in this case. So you are either capturing more or less of them, so signal to noise ratio may be affected. So also it's also not a very good idea to always want your signal to noise ratio to be perfect, because when you increase signal, you suppose are increasing your spatial resolution and everything. So in that case sometimes your noise also increases, because suppose you are for example FOV, if you go for a small FOV, you have small region of the brain that you are scanning. Compared to that, if you scan for a whole brain, you are thinking that yeah, I am taking the whole brain, so I am having more information. Along with more information, your noise will also increase, because there will be different kinds of artifact, motion artifact will get involved, there will be eye blinks. If you scan the whole brain, you will get more eye blinks, different kinds of artifact from your visual cortex also. So these are the things that you need to make sure that you know what it is, why you are doing it, what is your hypothesis. That based on that you will be basically deciding on what TRT ratio, what flip angle, what FOV or slice thickness is of your interest. And also there is something of voxel size, usually kept between 2 to 4 millimeter. If you increase the voxel size, again your signal to noise ratio may like go down, because this voxel size is increasing, but at the same time more and more artifacts may get involved in. Voxel is basically when your when you go inside the scanner, the scan is basically yeah. I actually so she was working on a project and she told me that you can you can collect some data. Participant will be going in as for Shashwat study, so he's explaining to him what he has to do inside. Yes, that's what Shashwat is currently doing. I am basically studying the neuroplastic changes in, first of all, I studied the neuroplastic changes in normal people as a function of music and motor training, because I'm trying to develop a neurologic music therapy for PD patients. So I wanted to make sure that the kind of plasticity that is developed for music training actually works for motor functions. So that's one thing I did. Now I'm doing it with PD patients. Yeah. हाँ, माधव का हाइट आपने देखा नहीं है, विल सी. हाँ, उसका हाइट के हिसाब से उसका वेट ठीक ही है सर. हाँ, माधव इज हियर. माधव इज वेरी टॉल. माधव एक बार आ. Show Divakar sir how tall you are. He is not being able to believe it. So. He is fine. He wouldn't go inside the machine. So.
But magnet is on na super.
No, but that much you can handle.
The belt is not of good quality.
होना तो नहीं चाहिए, but ठीक है. He is doing it for a long time. So I'm sure he has got used to it. But yeah, it does actually. Sorry. Yeah, the head coil has to go on when he is inside, because that head coil will actually first of all, it will give you, it will set the axis to 0, 0, 0, the red dot I told you that it will go on the forehead only, then he will send him in. So you know you are starting from 000, and also the head coil is because otherwise the participant will move their head a lot, and more motion artifact and eye blink artifact will get involved. So to remove that also head coil is there, and also to generate the contrast. That is how you will get the magnetic susceptibility controlled when you are doing the entire scan. So you have to maintain the magnetic susceptibility at a particular point when you're doing the scan. It should not change as much as it is changing at, like around your. Yeah. So there are different types of head coils. You can choose on the basis of how many number of channels and all you can change, but that is the main head coil. So person suppose they suppose use specs, so they are they have gone inside, and now we are doing a study which involves visual stimulus to which he has to respond. So that person suppose cannot see without the specs, so then we give them the goggles. Other than that you don't. It's balanced with what your power is, so the goggles that will be given to the participant, it will be balanced with that. But the headphone is like for everyone. It's for the noise cancellation that is already inside, because it's a magnetic field, so it will make a lot of noise once you start.
Music can also be.
Yeah, music can be improvised, but the problem is for that you will separately need a noise cancellation headphone, because they're already distressed with all the sound that is inside. So if you want to suppose do a auditory intervention or auditory stimulus, in that case you need a separate noise cancellation headphone. So. What exactly do you want to know?
Like I want to do for Alzheimer's, so if I know what to do for Parkinson, so I can think in such a way how I can do for Alzheimer's.
So
I don't know
it's normal. So for normal participants, the protocol you will put is the same protocol that will go for any patient population as well. So it's not like that if a person has Alzheimer's or Parkinson's, you will include or introduce any other kind of parameters in your fMRI protocol. The protocol will be same. The protocol is for everyone. It's for healthy controls and also for people who are like not healthy. They can have some motor problems, memory problems, like you were saying, Alzheimer's. So that is the head coil. You can see he has put on the head coil. Yes.
So it's the one with the red.
Yeah. The red dot has to. So that when you go inside, you can see. So it's put on here. Yeah. After it is put on here, he will send him inside the scanner, so that you start at baseline. So that is basically defining your baseline, that I'm starting at x is equal to 0, y is equal to 0, and z is equal to 0. Normal medical so basically if you are doing it in a medical setting, usually a brain scan cost these days around 12 to 15,000, and if you are doing other parts, I'm not very sure. This 3 to 5. Yeah, sure. I think 3 to 5. That varies depending on what you do, whether it's functional, you are using it for 1 hour. हाँ शाश्वत
अरे अभी तो आप बताओ ना, ऑर्गेनाइजर है, स्ट्रक्चर है, सब बताओ।
नहीं मतलब I have already told them DTI functional structural. I have already told them. हाँ. That's why I do it first and then start, so they understand. So first he will run a localizer. A localizer is basically, like I told you, when the red dot is here and the head coil is put on, you are putting the baseline as in x equal to 0, y is equal to 0, and z is equal to 0. Localizer basically localize your brain in 3 axes. So see, the image has come. So this is what the localizer does, that okay, one person is inside, it's not a phantom scan. It's a full head scan. I don't know. He will tell me. I have told him, but he is asking if is there a specific region. It's a whole brain scan, right? So in this case, the like field of view is the whole brain. You can choose based on what you want to do. Like he's doing motor learning, right? So if you have a particular like interest in motor learning, you can go for your basal ganglia or cerebral thalamocortical network. So those are the things you can look at. So see, I'll just show you. Can you see the yellow line up and down? See, it covers the whole brain. If you have a, suppose, FOV, you can change it.
How many slices?
He knows. How many slices?
Slices, right? I have
And what is the thickness?
2 to 2 mm. 2 mm.
So here it is defined already.
So there is
It's entirely there.
the slice thickness will be 2, and then you can see a distance acceleration factor. I think mine is on 20 or yeah, it's 20. So 20% of 2 will become 0.4. So between every slice there is a 0.4 mm gap. That is the distance acceleration factor.
The distance between the slices, right?
How we decide slice thickness?
You can see from your whatever papers you have previous papers, but generally what happens is that in old papers you'll find like they have 3 and 3.5.
So that's why I said that if you keep it thinner, you will get more better spatial resolution, but it will take more time. But 2 mm is fine.
2 mm is the
Yeah, but you should not go for like 4. It's very thick. It will be done in very small amount of time, but you will not get very good spatial resolution. It will be very blurry. Yeah.
Structural is
1.
You can go for
0.8.
For functional, lowest I have seen is 1. But 2 is fine. So whatever you are using, you can use FSL, AFNI, SPM. It's fine. Yeah.
So Voxel size 0.5 to 3, कभी 1.
Voxel size usually is kept at 2, but it can be 1, 0.5. But 2 is standard. Like so, why 2 for everything? Because it's at a point where it will not affect your SNR much. So
Voxel is a cube. So when we say voxel size, what is it?
Is the X, Y, Z. So
2 to 2.
Yeah, it's written as 2 cross 2 cross 2. Yeah.
So how many is supposed to see inside?
This is 9 minutes. कितने time का है शाश्वत?
structural.
Like she's asking how long will he be inside?
The task is only for 5 minutes. That is only for the demo.
This is a total of 15 minutes, but like so it varies for person to person and their task and the other things they have. For example, suppose I have a DTI, my entire protocol is 20 minutes. It has a localizer, 1 resting state and 1 DTI. This is total 15 minutes.
Yeah, total 15 minutes.
Total
task.
15 minutes. But tasks can go up to 1 hour where they have to keep doing. So if you are studying a particular behavior and the neural mechanisms behind it, you have to do it again and again to validate that okay, this is the region for that particular behavior. कौन सा फोल्डर?
Previous tasks.
सब हट गया क्या? ई प्राइम में है शाश्वत, सुबह ही तो देखा मैंने. सुबह दीपाना के चलाया अपना. हाँ बार्ट वाला चलाया था सुबह.
So अभी कैसा सर उसका FOV चेंज किया था.
This is structural. This is the question.
Orientation.
Yes. So it doesn't mean that the number of slices or the region you are covering has changed. It's the way you are, like the sagittal, coronal, radial. It's that kind of change in terms of. So it's not like changing axis wise, but he is just making it the rotation here. Yes. So that it gets more clear in. So you will do whatever is needed to get as good spatial resolution as you can. So see, when you analyze, you will run multiple filters to remove this, that, right? So make sure what you take from here is already not contaminated. That's the key takeaway. So if you make a lot of changes and all that, you can introduce a lot of errors. So he is just trying to capture whatever is the best spatial resolution. Because yes. Because if you go for FSL, SPM and all that, we use, there are different filters to remove. So what kind of errors do we get in the fMRI? One type of error is coming from the participants themselves, eye blink, head movement, right? Another type of error comes from the machine, because there's a varying magnetic field. So there is magnetic susceptibility. If there is varying magnetic field, there is also a varying electric field. There is eddy current. So these you have to remove.
This noise that is coming from the scan,
Mhm.
does that add to any sort of noise?
The noise that is coming from the scanner does no. So you are capturing the brain.
Basically, what I'm trying to say is, whenever the sound is being coming from the
Mhm.
machine, right?
Mhm.
Will that cause any effect or any activation in the auditory canal and then leading to auditory?
Yeah, then it can lead to auditory artifact, right?
Yeah.
So
That will be common for your baseline
as your task,
So it's going
because
to contrast, yeah.
yes, because it doesn't change in the baseline. It's the sound is there, in the task also the sound is there.
So in the resting state, as you're doing, that will be a constant noise also.
Yes. So suppose if you there is movie screening in resting state, and there is a situation where there is no movie screening, right? So you subtract which one, the baseline from the movie screening situation. In the baseline, the auditory artifact is already there. In the movie screening also the artifact is there. So the artifact does you, yeah.
And if you have auditory stimulus, anyways, they're hearing from the head. So while that is going on, while stimulus is going on, then they're not hearing the noise.
So that
machine noise is not audible if you are having it's not, it's that noise cancelling headphone. You can always have a better noise cancelling.
and also for auditory stimulus you have a separate noise cancelling headphone which you have to put on. That is not the headphone he has. Yeah.
Also earplug is given.
Yeah, earplug is given. So this is the earplug he is wearing for
Inside
now.
also there is
Inside also, if some people have tinnitus or any kind of auditory problems, that is given separately. So this is for just he doesn't get much affected by the noise. But if you are doing an auditory intervention or auditory stimulus based task design, you need a separate kind of noise cancellation. That cancels the external noise completely. So you will hear your stimulus and nothing else. Yeah, it's live. No, this is the.
So the T1 is complete. So you can see the T1.
Yeah.
That is the T1.
That is the T1. That the one you are seeing.
You're just seeing one slice, but you can see it once it comes.
Once like the entire thing comes, you can see the. Yeah. You can also see the 3D with the entire structural thing done. The 3D you can also see. He'll show you a holograph.
So the processing is done.
No. So you have to take can do in. So processing can be done on this also. There is a different software for it, but because someone is always collecting data, we don't keep this machine engaged in that. So we take it and we use our systems or our personal systems also to analyze it. Hey, you can see the scan timing.
Next to the scans, you can see the volumes also. I think it's 50, 58. Yeah. So out of 200, there will be 200 volumes out of that. 50 or 60 are done. Each volume will have volume slices.
So I hope things are mostly clear. Parameters and yes, please ask.
So we are conducting this experiment right now. Same experiment for maybe different stimuli or resting stimuli. If we perform with the same subject, so how likely we will get the
same result? So So you remember today in the lecture, in the first slide, sir talked about something called linear time invariant system, right? So that's a basic assumption of fMRI, that you are doing it, and if you do the same task with the same person n number of times, all the times your critical regions, critical activations of the regions will not change. So in fMRI there is something called smoothing. So whenever a region is activated or deactivated, the region which are close to it get some of those activation or deactivation, right? But the critical region, that is the, for example, suppose memory and hippocampus, there is a direct connection. Movement and basal ganglia, there is a direct connection. So these critical things, critical regions will not change. But yeah, when you are suppose doing the smoothing thing with your analysis, that may cancel out 1-2 different voxels every time you are doing, but the critical regions will not change. That is the basic assumption on the basis of which we are doing fMRI, that this system is like time invariant.
Across the screen, we can see the same.
Yes, yes.
But in between them, they won't show the similarity.
No. So what I am saying is, in terms of if you see the smoothing thing, so there may be some voxels which did not get the activation the first time, but they are getting it the second time. But in smoothing effect, they will be cancelled out. But the critical region that gets activated will not change if you do it n number of times. Same. दिवाकर सर वो holograph भी दिखाइए. The 3D. दिखाइए एक बार, अच्छा लगता है देखने में. Yes. It's called linear time invariant system.
For something for the functional outcome. So basically, in your functional scans, it's blurry, right? Because you're having a boxes of two and it's very fast. You don't care about the functional stuff, like the data. You don't care about the structure. Okay. So since everyone has a different brain structure and everything, you take a structure.
Yes.
The template can be applied to the functional ones. So instead of having like a structural scan in between your functional every time, you always have a template through the structural scan.
You overlay the.
Yeah, exactly. You just scale up every region according to the structural scan.
So only T1 is used.
Yeah.
Basically for overlaying.
For anatomy we use T1. For looking for anatomy.
Anatomy you said. Yeah.
So we do here contrast scales.
What is contrast scales? अच्छा.
डायग्नोस्टिक सेंटर तो है नहीं, तो इधर तो इधर लगा
Said all ना, we give like 2 tasks to 1 contrast injection fMRI, contrast fMRI.
Oh, अच्छा वो no.
पूरा सेटअप चेंज लगाना पड़ेगा ना.
आपने इसका
स्मार्ट ड्राइव ही चाहिए, उसके लिए बहुत काम पड़ेगा. वो तो बहुत चीज बढ़ जाएगा इससे ना.
So right now this is structural scales are
structural done, resting done. Now task.
task function. Yes.
Yeah, the function scan.
So what is our task?
I'll explain it here.
मोटर क्या है?
Learning. इसका आपने 002 रखा है.
हाँ जी. इनको ब्लर जरा दोनों के साथ दिखाएगा.
कौन? Okay, so my task is on motor learning. It's basically that you see 4 circles here. The black circle appear on any of the 4 locations. I've instructed him to press this key corresponding to whichever location is there. So there's 4 locations. Wherever so now it's appearing on second, he presses the second key. Similarly, right? So this is for the practice. After the practice, we have the main blocks. In that, it is in the entire sequence is in order. Also, the third location depends on the location of the first two, like the before two. So they come in triplets. But this rule is not known by the participant. So if they can figure it out, then we tell or we say that they have gained explicit knowledge of the sequence. If they don't figure it out, we say that they have not gained explicit knowledge. And if they have gained implicit knowledge, which they also don't know that they know the sequence, that we can see from the reaction times. So if the reaction time has been decreasing per block, then we say that they have implicitly learned the sequence. But if you ask them to generate the sequence, give the order or predict the third location, then they won't be able to do that. Sir, wo S-Pulse wala. Sir, S-Pulse.
So you take a subjective reporting
No.
after the experiment is done?
We take a free generation. We can take a free generation. In free generation, you just tell them to press 12 random buttons or press buttons in which they think the sequence happened. So they press 12 buttons and then you determine based on what is the longest length of they could reduce. Yeah, the correct sequence. And otherwise, apart from free generation, you have different tasks in which one of the tasks is called inclusion task. In that, what you do is you show them 2 targets or 2 locations and you tell them to predict the third location. And the other one is you show them 2 targets and you tell them what is the which location is the impossible location to happen.
Scan is running, we didn't give any task, we didn't inform, or we informed the subject.
We informed them, no?
Earlier.
Yeah, I've told them. I've given him instructions, everything. So the only thing is that the triggers which we have, it's like a pistol. So you have a left thumb, left index, right thumb, and right index. The keys corresponding to are A, B, C, and D. So if they press the right thumb, that is equivalent of me pressing A from here. And similarly for B, C, and D. Now in my task, since I have 4 locations, what I've told him to is just put it horizontally, and then this corresponds to the first location, second location, third and fourth, and then he can press according to whatever he's getting. The ideal way would be to get a trigger box which has all 4 responses in horizontally. Yeah.
So now he is clicking.
Yeah, he's clicking. So every beep you get, that's a sound, and this corresponds to A, B, C, and D. So yeah, it's inverted, so you won't be able to match it properly. But if it was A, B, C, D, then you could see that he's pressing A, B, C, D according to the location. For right now, for him it would be D, C, A, and B, since it's like this. So D, C, A, and B. Now if you can map it out, like process it that fast, then you can see. Yeah. So I was coming to that point. Yeah. So since for me it's a learning task, and so the design, whether you want to use a mixed block or a event related, will depend on what you're looking at or what the activations you would want to analyze. In my case, learning is happening even when the stimulus is not there on the screen. So he's pressing. So basically, these are just images. It's not a a video. So he's pressing, and then the image disappears, and the next image comes up. So in an event-related design, what you have is wherever the image is coming or the stimulus is coming, you analyze only the activations which are there for that stimulus, right? In my case, you can see the blank screen, right? So once he presses, it's blank, and then the next stimulus is coming. Even in the blank, the space which we call response stimulus interval. So even in that interval, they are still learning the sequence, right? Or they're trying to process it. They're trying to get to the sequence or whatever. It's implicit. They don't know that they're doing it, but they're doing it, right? So I would want sustained activation rather than what is the activation only for that event, right? So in my case, I'm using a block design specifically because of that. Now, when you're using a block design, you'll need to specify the duration of the block. In my case, this is 36 seconds. You wouldn't want a block more than 40 seconds or 1 minute. You have seen from today's lecture also, right? So ideally, a block should be between, ideally it should be from 20 to 25 seconds. If your task in my case For me, the reason why I say that it should be between 20 to 25 is that generally in block designs you just have one stimulus which is appearing for 20 seconds. In my case, the stimulus is changing. So in my case, the optimal duration will be different. But since I don't want to take any risk, I have it below 40. If you have one stimulus appearing, don't go beyond 25. Have it like 15 to 25 is the optimal range, right? And so my sequence is that I have block, then a block for 36 seconds, a rest period for 12 seconds, a 36-second block again, 12-second rest, and so on. After 4 blocks, I have a control block that will act as a baseline for my previous blocks. Now the control block is that he's just pressing one key. For the demo, because I'm showing it only for 5 minutes, I have only one control block, so he's going to press the right thumb key every time. So he's going to press that for 36 seconds. Oh yeah, that's happening. Yeah. So it's stationary. So this is the control block. Now I'll contrast this with the other one. So I'm getting a motor response as well as a spatial activation from it. So I can minus that and I can get only the learning part from it. So you need to design your baseline according to your task. Your TR, your TE, everything depends on your task.
So we can check IQ also.
If you can design a task in which they are already responding to, you give them 4 options, like option A, option B, option C, you can do it. But depends, why do you need fMRI for that? That is the main question. Yeah. If you really want to see like what's happening, how easy, like IQ, like suppose RPM, Raven's progressive matrix, in that you can have it for like a pattern recognition task or something like that. Yeah. So it depends on the question if you need fMRI or not. We have a task in which we have taken rating also, but the only problem is that you can give only these 4 options, or you can go about a way in which you give like a text and then they can increase or decrease the value until they come to the desired value. So you can give a rating, it's just that you have to code it out. That's the only part. If you have 4, it's very easy. Just tell them press A, B, C, D. Yeah. You can't see it live because this is the last protocol. If we had